This video is an exploration of the catalytic mechanism of chymotrypsin and the experimental results that support it. Chymotrypsin is one of the most studied enzymes, and since it uses multiple types of catalysis, is commonly used as an example mechanism in introductory biochemistry textbooks. Studies of the enzyme's mechanism began in the 1930s and early 1940s, predating the advent of molecular biology. Therefore, the chemists studying the chemical function of the enzyme relied on chemical techniques. It was one of the first enzymes to have been studied by X-ray crystallography, which provided additional details on the mechanism. All the techniques used to study chymotrypsin are still valid today, even though they might not be our first choice since we now have many additional techniques available. This is a reminder that this presentation is Dr. Johnson's intellectual property. Chymotrypsin belongs to the family of enzymes known as serine proteases. We will begin with the structure and function of the enzyme. I recommend that you be familiar with enzyme classification and the general function of enzymes, enzyme kinetics, and enzyme inhibition before watching this video. If you need a refresher, I have video series on these topics. Links to them are in the show notes below. Chymotrypsin is one of the enzymes that allows us to digest proteins in our diet. It was discovered in the early 19th century and was crystallized in the 1930s. The name chymotrypsin does not follow the nomenclature used for most enzymes, since it does not have the ase suffix. This is because this enzyme was well known before we started naming enzymes with this suffix and before systematic nomenclature of enzymes was developed. This enzyme has the EC number 3.4.21.1, indicating that it is a member of the hydrolases that act on peptide bonds, that is, they have peptidase activity, and belonging to the group known as serine endopeptidases. It is the first member of this group. Endopeptidases are enzymes that cleave the peptide linkage between residues inside the peptide chain, as opposed to at the end of the chain. This bond is known as the sessile bond. Among the other members of this group are 3.4.21.4 trypsin, 3.4.21.5 thrombin, 3.4.21.11 elastase, 3.4.21.20 cathepsin G, and 3.4.21.62 cetylysin. Chymotrypsin is synthesized ribosomally in the pancreas and forms 5 disulfide linkages post-translationally. This is a zymogen, or proenzyme, a precursor form of the enzyme. It is not yet active. This is important so that it does not digest the pancreas. The zymogen is secreted into the small intestine, where it is cleaved by trypsin. Further autolysis, cleavage by itself, yields various forms of the active chymotrypsin enzyme, alpha, pi, delta, and gamma. Of these, alpha chymotrypsin is the predominant form, and the one on which most research has focused. The mature or active enzyme has 3 chains. The A chain, residues 1 to 13, is 13 residues long. The B chain, residues 16 to 146, is 131 residues long. And the C chain, residues 149 to 245, is 97 residues long. Although alpha chymotrypsin is a monomer in solution, it crystallizes as a dimer. In this view of the enzyme, you are seeing just a monomeric unit. The alpha chymotrypsin structure contains alpha helices, beta barrels, and surface loops. The active site is in the cleft between the 2 beta barrels. The combination of the 2 6-stranded Greek key beta barrels and 8 surface loops that surround the active site is a characteristic type of protein fold known as the trypsin fold, which is found in related serine proteases. One of the features that distinguishes chymotrypsin from other digestive endopeptidases is the location of the peptide bond that it cleaves, also known as the sessile bond. This is primarily after the large nonpolar hydrophobic residues tryptophan, tyrosine, and phenylalanine, and secondarily after smaller hydrophobic residues, especially leucine and methionine. Cleavage can also occur after other nonpolar residues, but this is very slow in comparison, and the resulting specificity constants are several orders of magnitude smaller. The nature of the residue on the other side of the cis-al bond appears not to affect the rate of substrate binding. Now that we know that chymotrypsin hydrolyzes the peptide bond, we'll have a look at the chemistry of peptide hydrolysis. The peptide hydrolysis reaction has several challenges or obstacles. Overcoming these is crucial for making the reaction more feasible. First, the peptide linkage is stable due to it being a poor electrophile. The peptide bond is planar and rather rigid because of significant resonance contribution originating from the lone pair of electrons on the nitrogen atom. This reduces electrophilicity of the carbonyl group, making hydrolysis of the peptide bond relatively slow and difficult. Secondly, water is a weak nucleophile. Thirdly, the amine is a poor leaving group. Under neutral conditions, a typical rate of hydrolysis is around 10 to the minus 11 seconds to the minus 1. Another way to look at this is that it would take about 31,500 years for a single peptide bond to be hydrolyzed at neutral conditions without any catalysts. We find that the reaction proceeds faster below pH 4 and above pH 10, where either acid or base catalysis become more important. In this diagram, the two orange lines indicate the extrapolated rates of hydrolysis under acid or base catalysis. These overlap in the middle, causing the actual results to level off. There are therefore two non-enzymatic conditions for catalyzing the hydrolysis of a peptide, or amide in chemical parlance. In the acid-catalyzed reaction, the carbonyl must become protonated to increase the electrophilicity of the carbonyl. The more acidic the reaction conditions, the more concentrated the protonated carbonyl species will be. The water nucleophile attacks the electrophilic carbonyl carbon, causing the pi bond to the protonated oxygen atom to break. The extra proton on the oxygen that came from water is removed by solvent. This neutral species is not quite ready to finish the reaction. The nitrogen of the peptide is not a good leaving group. If it leaves right now, it will have a negative charge and be very unstable. It can be made into a better leaving group if it is first protonated. This proton will come from the pool of protons associated with the acidity of the solvent. Formation of a new carbonyl between one of the two oxygen atoms and the former carbonyl carbon atom forces out the protonated nitrogen leaving group, resulting in the formation of a protonated carboxylic acid. This species undergoes deprotonation of the carbonyl oxygen and then deprotonation of the carboxylic acid to yield the final carboxylate and organic ammonium ion products. In the base catalyzed reaction, hydroxide is the nucleophile. Because it is a relatively good nucleophile under aqueous conditions, it can attack the carbonyl of the peptide without first needing the carbonyl to be protonated. The negatively charged intermediate can reform a carbonyl and push out the nitrogen leaving group, which, being a strong base, removes the acidic proton from the carboxylic acid so that we form the final carboxylate and amine products. Given these entirely chemical catalysis mechanisms, how does chymotrypsin accomplish the hydrolysis under relatively neutral conditions? Let's look at the evidence that was collected to establish the catalytic mechanism of the enzyme. For any type of kinetic study, we need to have a way to detect and quantify either formation of product or loss of substrate. Early studies did not have good spectroscopic methods for doing this. By the 1950s, researchers were looking at many different possible substrates, particularly ones that yielded chromophoric products that could be detected easily by spectrophotometers. Paranitrophenyl acetate fits the bill. Hydrolysis of the compound yields a yellow anionic product that is easily detected spectrophotometrically at 400 nanometers. It is actually a poor substrate for the enzyme since it lacks a large hydrophobic group in the alpha position. However, since it is an ester rather than an amide, and esters are more reactive than amides, it is hydrolyzed more quickly than the corresponding amide would be. This turned out to be important to understanding the catalytic mechanism. When researchers followed the initial reaction rate of chymotrypsin action on paranitrophenylacetate, they found that they routinely obtained biphasic curves like this one. The curve you see is actually the combination of two reaction curves. The first part is an exponential reaction that gets cut off by a later linear reaction. Normally, we would expect this linear phase to show a small increase in absorbance with time, as illustrated with the orange dashed line. However, here it drops down instead. How can this be explained? The spectrometer used had some electrical drift, and the liberation of protons from the reaction neutralized the paranitrophenolate ion. The neutral paranitrophenol does not absorb light at 400 nanometers. When the linear phase is extrapolated back to time zero, the resulting value correlated exactly with the amount of enzyme used in the reaction. In other words, if they changed the amount of enzyme used, this value would change accordingly. The two-phase reaction that is dependent on enzyme concentration is evidence for the formation of a covalent intermediate with the enzyme, as one would expect to find in a double displacement kinetic mechanism. The idea of an acyl enzyme intermediate was already proposed for another enzyme, acetylcholinesterase, by 1950, so it was not much of a stretch to apply the same concept to chymotrypsin. In fact, the acyl enzyme intermediates that can be formed by reaction with various esters were carefully isolated. These acyl enzyme species could not catalyze the hydrolysis reaction, but removal of the acyl group rendered the enzyme catalytically competent. It was not possible to isolate the acyl enzyme intermediate from reactions in which an amide was used as the substrate. The kinetics for the formation of a covalent enzyme intermediate are described in this kinetic reaction scheme, where E'(S) is the acyl enzyme intermediate. For substrates such as peptides, in which the formation of the acyl enzyme intermediate is the rate-determining step, the effects of nucleophile strength and leaping group ability are observed. The rate of decomposition of the acyl enzyme intermediate is not observed because it occurs after the rate-determining step. For substrates that are more easily hydrolyzed, like esters, formation of the acyl enzyme intermediate occurs before the slowest step, hydrolysis of the acyl enzyme intermediate. Here, the nucleophile strength and leaving group ability of the substrate are not significant to the overall rate of reaction. At this point, we have established a kinetic mechanism involving an acyl enzyme intermediate. The next question that was addressed is, what is the nucleophile? After diisopropyl-fluorophosphate was found to inactivate acetylcholinesterase, researchers decided to see what it would do to chymotrypsin, since chymotrypsin has esterase as well as proteinase activities. They discovered that, like with acetylcholinesterase, it inactivated the enzyme in a time-dependent manner, as did other related fluorophosphate compounds. Furthermore, both the esterase and proteinase activities of chymotrypsin were affected at the same rate and to the same extent. The amount of DIPF required for complete inactivation was stoichiometric with the amount of enzyme in the assay. Therefore, they concluded that these activities relied on the same active site and that there was only one active site per molecule of enzyme. For both enzymes, the activity could be regained by treatment with hydroxylamine. The inactivation and subsequent recovery of activity after treatment with hydroxylamine suggests formation of a covalent bond between the inactivator and specific amino acid residues in the enzyme's active site. Inactivation of the enzyme with radiolabeled DIPF, followed with careful digestion and isolation of the label, revealed that the residue that reacts with DIPF is serine 195. Around the same time that researchers found that chymotrypsin could be inactivated by reaction with phosphate esters, they also observed that the reaction intermediate we noted on previous slides could be isolated at about pH 5. This acetyl enzyme is formed in stoichiometric quantities and is catalytically inactive but unstable. It can be reactivated by reaction with water at slightly alkaline pH or treatment with hydroxylamine. Reaction of chymotrypsin with sterically bulky esters, such as paranitrophenyl trimethyl acetate, yield an acyl enzyme product that is stable enough to be crystallized. However, this acyl enzyme decomposes after repeated recrystallization or storage, likely due to slow reaction with trace amounts of water. Further evidence of the acetyl enzyme is that it can react with alcohols to yield an acetyl ester, effectively completing a transesterification reaction. Researchers at the time found that transesterification is faster than hydrolysis. The chymotrypsin liberated in this process is fully active. Researchers did not identify what they called the specially reactive group in the protein as serine from these studies because the acyl enzyme structures are not sufficiently stable. However, they did note the similarities between these inactivation reactions and those with DIPF, and also that the denatured enzyme was not capable of reacting with the acetylating substrates of DIPF. Although the environment inside an enzyme's active site is more hydrophobic than the cytosol, this environment does not seem to be sufficient to make the serine hydroxyl group a strong enough nucleophile to account for the rate acceleration that the enzyme can achieve. Therefore, there must be other residues involved in catalysis. One of these was identified by affinity labeling. Tosylphenylanilyl chloromethylketone, or TPCK, has a particularly electrophilic alpha carbon. The adduct resulting from reaction with the enzyme inactivates the enzyme and is stable enough to be isolated. Acid digestion of the inactivated enzyme revealed that the reactive residue was histidine, and sequencing indicated that this reactive residue was histidine 57. No other histidine residues reacted. These results are consistent with earlier work on the pH rate profile of the enzyme and X-ray crystallography work done later. pH rate profile studies showed a bell-shaped curve for the acylation of the enzyme under conditions in which the deacylation reaction is not rate limiting. This curve has an inflection point at about pH 7 and a second inflection point at about pH 9. Under conditions where the deacylation is rate limiting, the curve is sigmoidal with only the inflection point at about pH 7. This inflection point suggests the involvement of a histidine residue in the catalytic mechanism and is in agreement with the affinity labeling results we discussed on the last slide. In the late 1960s, chymotrypsin was crystallized and it became the first enzyme to have its structure determined by X-ray crystallography. The crystal structure showed the active site serine and histidine residues, as well as other residues that interacted with the active site histidine. Initially, the main residue that interacted with histidine was identified as asparagine. This was puzzling because it was not clear how an asparagine side chain would interact with the histidine side chain. It was challenging to distinguish between asparagine and aspartic acid residues by the sequencing methods used at the time. Once it was determined that the identity of this residue was incorrectly determined and that it was in fact aspartic acid, it became apparent that the aspartate side chain was helping to offset the positive charge the histidine would acquire upon removing a proton from the serine hydroxyl group. By 1969, the Blow group described this effect as a charge relay system. In the mid-1990s, other chemists argued that the interaction was due to a low barrier hydrogen bond. This topic has been hotly debated for many years. Regardless of whether this is a charge relay system or the effect of a low barrier hydrogen bond, The combination of the three residues, serine, histidine, and aspartate, is known as a catalytic triad and is responsible for the nucleophilicity of serine and much of the rate enhancement provided by the enzyme. The crystal structure of chymotrypsin also revealed two other pieces of information that help us understand how this enzyme works. These are more clearly seen in crystal structures with the enzyme and an inhibitor. First, there is a pocket in the active site that binds to the oxygen atom of the scissile peptide bond. This pocket is known as the oxyanion hole. Interactions between the amino groups for the backbone of residues glycine 193 and serine 195 that line the oxyanion hole, and the substrate's carbonyl oxygen serve to make the carbonyl more electrophilic and stabilize the negatively charged tetrahedral intermediate. Like we saw for the catalytic triad, the oxyanion hole also contributes to the rate acceleration accomplished by the enzyme. The second pocket of interest in the active site is the S1 subsite, which is responsible for the specificity that chymotrypsin has for large aromatic residues to the N-terminal side of the scissile bond. This pocket is relatively wide and nonpolar, corresponding to the large nonpolar hydrophobic residues that bind best. Small and charged amino acids just don't fit well in this subsite. At this point, we have established that chymotrypsin cleaves the peptide bond just to the C-terminal side of a large, nonpolar hydrophobic residue. We saw that it operates with a double displacement kinetic mechanism involving an acyl enzyme intermediate. We have identified the residues of the catalytic triad, serine, histidine, and aspartate, and the presence of an oxyanion hole to stabilize the tetrahedral intermediate. It's time to look at the catalytic mechanism. The first substrate, a peptide or protein, binds in the active site with the S1 specificity pocket and oxyanion hole placing the scissile bond near the nucleophilic serine of the catalytic triad. Interaction of the scissile carbonyl group with the oxyanion hole increases the electrophilicity of this group. The serine hydroxyl group attacks the carbonyl group, pushing the pi electrons onto oxygen. As the serine hydroxyl group is attacking, the imidazole ring of the histidine hydrogen bonded to it acts as a general base to remove the proton from the serine oxygen atom. The developing positive charge on histidine is stabilized by the aspartate side chain. The substrate's tetrahedral intermediate collapses to reform the carbonyl. Either the serine oxygen or the peptidyl nitrogen will serve as the leaving group. The leaving group takes the proton from the protonated histidine side chain as it departs. If the serine oxygen is the leaving group, no reaction is observed. However, if the peptidyl nitrogen is the leaving group, the scissile bond will be cleaved, resulting in the acyl enzyme intermediate. Since nitrogen is more basic than oxygen, the nitrogen atom is more likely to be protonated. The first product, the amino product, leaves the active site and is replaced by the second substrate, water. Now water functions as the nucleophile. It is a poor nucleophile, but deprotonation by histidine, assisted by aspartate, increases its nucleophilicity. This forms a second tetrahedral intermediate, which is again stabilized by the oxyanion hole. Note the water is reacting with the more electrophilic ester, the acyl enzyme intermediate, instead of the initial amide or peptide substrate. Collapse of the second tetrahedral intermediate to reform the carbonyl group results in cleavage of the bond to the serine oxygen atom, which gets reprotonated by the protonated histidine residue. The chemical reaction is complete. The second product leaves the active site, returning the enzyme to its resting state so that it can begin a new cycle of catalysis. This mechanism for catalysis accounts for the observations on the activity of chymotrypsin. The kinetic mechanism and the observation of an acyl enzyme intermediate, the catalytic triad of serine 195, histidine 97, and aspartate 102, and the pH rate profile indicating histidine needs to be able to act as a base. Instead of using hydroxide or the hydronium ion to catalyze the reaction, the enzyme achieves rate acceleration of around 10 to the 10 times greater than the uncatalyzed reaction using a combination of strategies, including both general base and general acid catalysis, nucleophilic catalysis, and electrophilic catalysis. Did studies of chymotrypsin stop once its catalytic mechanism was established? No. Modern tools have been applied to chymotrypsin to gain even further insight into how it works and to understand the relationships between it and other enzymes catalyzing similar reactions. Chymotrypsin was one of the first enzymes to have been studied and to have its catalytic mechanism established. Because of the time in which experiments crucial to our understanding of the function of this enzyme were performed, some of the tools that are commonly used today were not available. So, how would we study the mechanism of chymotrypsin if it were a newly discovered enzyme? Using molecular biology tools, we would determine its sequence, either by sequencing the protein or converting the gene sequence to an amino acid sequence. Using the resulting sequence, we would do some homology searches and alignments to determine if there are any similar enzymes whose function we do know. This would be a starting point for testing out possible substrates and inhibitors. We would use site-directed mutagenesis to establish that the catalytic residues are also essential in our new enzyme. We would want to know the structure of the enzyme. If we can obtain diffracting crystals, we could use X-ray crystallography to obtain the structure. Alternatively, we can use cryo-EM to determine the structure of the enzyme without needing to crystallize it. One of the successes of AI is the development of AlphaFold, which can be used to predict a 3D structure of the enzyme. Once we have generated a 3D structure of the enzyme by one of these methods, we would compare this to existing structures of related enzymes as well as others to look for both similarities and differences. We could decide to study the enzyme's structural dynamics to understand the conformational ensemble and how the structure of the enzyme relates to its activity and recognition of substrate. In fact, some scientists are already studying chymotrypsin and related proteases in this way. Since site-directed mutagenesis became possible, scientists have applied it to study other serine proteases, including trypsin and cetylysin. Changing either the catalytic serine or histidine residues to alanine dramatically decreases catalytic activity. Catalytic activity is also lost if the active site serine is converted to threonine. While changing the active site aspartate to either asparagine or alanine also results in a loss of activity, this is not nearly as much as for the active site serine and histidine residues. These results confirm the roles that we established for these residues in the catalytic mechanism. The activity of the mutant enzymes are not lower because the catalytic triad is not the only contributor to the overall catalytic power of the enzyme. The oxyanion hole, desulfation, and the nature of the conformational ensemble are all thought to contribute to the overall rate acceleration. How is chymotrypsin related to other enzymes? Based on structural information and sequence homologies, serine proteases using the ASP-His-Ser catalytic triad are believed to have evolved at least 4 times. Chymotrypsin belongs to the trypsin-like family of serine proteases, which includes trypsin and elastase. All the enzymes in this family have the trypsin fold of 2 Greek key beta barrels. The cetylysin-like family has a 3-layer sandwich structure. This family includes cetylysin, aromatase, and alkaline protease. The SC peptidases use an alpha-beta hydrolase fold structure and include prolo-oligopeptidase as well as enzymes with esterase activity, such as the well-studied acetylcholinesterase. The SK peptidases have an alpha-beta structure. The representative member of this family is CLP peptidase. One group of serine proteases use a glutamate residue instead of the aspartate found in the canonical catalytic triad, while other serine proteases use other combinations of amino acid residues to increase the nucleophilicity of serine. The serine proteases are just one catalytic mechanism for achieving peptide hydrolysis. Aspartic proteases use one or more aspartic acid residue in their active sites to catalyze peptide hydrolysis. Examples of these include cathepsin, renin, and pepsin. Some enzymes use glutamic acid residues to achieve this activity. These are known as glutamic proteases. The cysteine proteases use cysteine as the nucleophile. Papain, caspase, and the hepatitis C peptidase 2 all use this strategy. Some of the cysteine proteases use a catalytic triad similar to the trypsin serine proteases and could be structurally related. Threonine proteases use threonine as the active site nucleophile. Metalloproteases use a metal ion, usually zinc or cobalt, to activate the water molecule. Some examples include aminopeptidase N, thermolysin, carboxypeptidase A1, and anthrax lethal factor. Additionally, enzymes that function as hydrolases might use a catalytic triad. Examples of these include asparaginases, esterases, acylases, and beta-lactamases. In this video, we have discussed the enzyme chymotrypsin. We started by looking at its EC classification, zymogen maturation, and structure. Then we looked at the substrate specificity and catalytic challenges the enzyme needs to overcome and reviewed non-enzymatic peptide hydrolysis. We discussed some of the evidence supporting the catalytic mechanism, including the acyl enzyme intermediate, roles of serine 195, histidine 57, aspartate 102, and the oxyanion hole. Then we examined the catalytic mechanism of the enzyme. Finally, we talked about other ways to study the enzyme. If you found this presentation useful, please like, comment, share, subscribe, and push the notification bell for my next upload. These actions will help other students find my content.